So again, thank you all for being here. Today I'm going to be talking about my PhD research, where I used a technique called Lcms Metabolomics to study the chemistry of members of the hops of the species Cumulus. And so this is broken up into 3 separate projects, one dealing with wild hops, species Cumulus neomexicanus, and then a drought study on hops, and ending with a terroir study, which I'll explain what that means when we get to it. So before I get into the meat of the talk, so this talk and my dissertation are dedicated to my parents, who are on the call right now, and you know, I really couldn't have done any of this without their support. They've given me, so many opportunities to succeed, and all of my success is thanks to them. So this talk and my dissertation are dedicated to my parents. So here's the outline of my talk. I'm going to start with some background information, and then I'll talk about each one of my projects in turn, and then just end with a few major conclusions of my work, and then end with acknowledgements. So first, the background information. So what are hops? So hops are referred to plants in the species Cumulus, which is in the botanical family Cannabaceae. So hops are perennial, twining vine plants, and they're dioecious, which means that the male and the female flowers occur on separate individual plants. So the male flowers you can see down here are sort of smaller, and the female flowers are here, they're bigger, and these are actually, they're called inflorescences, they're compound flowers. So each one of these structures actually contains multiple flowers. And these flowers also called hops. In English, in German it's Hopfen, Turkish Şerbetçiotu, in Greek it's Lykiskos, and in Chinese it's Jiuhua. So this is the part that's most often used by humans, and by far the economically most important part of the hop plant is the female flowers, also known as cones, which produce a wide array of phytochemicals, plant chemicals that are useful for humans. And the leaves, which are also, 2 of my projects are looking at leaf chemistry, and one of the projects is looking at cone chemistry. So the cones are the economically most important part. The leaves produce a lot of the similar chemicals to cones, and studying leaf chemistry can be useful for understanding the biology of the plant, what's going on biologically and physiologically. So this is the hop plant. As I mentioned, the cones are economically the most important part, and by far the most common use today of hops is in beer. So due to their specialized chemistry, hops impart bitterness and aroma to beer. So nowadays they're most often associated with the bitter flavor of beer, and it's thought that originally hops were added to beer as a preservative to prevent spoiling, because some of the compounds in hops inhibit bacterial growth. So hops today are most commonly used in beer. Hops also have medicinal value, and one of the common medicinal uses is to make a tea out of the cones, which you can see here. And the tea has a sedative effect, which some people take to help with sleep. But by far the most common use of hops is in beer today. So as I mentioned, hops are in the family Cannabaceae. And the other important genus in that family, in the Cannabaceae, is the genus Cannabis, which contains cannabis, marijuana, hemp. So hops and cannabis have a lot of similarities in their chemistry and the chemical compounds that they produce. Although I should point out that hops do not produce the cannabinoids, which are sort of the psychoactively important and active constituents of cannabis. And I'm not going to go too much more into detail about cannabis in this talk, but I did think it was worth mentioning. So one of the sort of central questions that all 3 of my projects address is this question of how to genetics and environment affect hop phytochemistry. And I'll explain what that means on the next couple of slides. So to do that, I'll first turn to an example of apples, another crop, which is perhaps more familiar to a lot of people. So here we have apples from many different varieties. And now just by looking at them, you can see that they have different colors. And the different colors are characteristic of that particular variety or that particular genotype or genetic group. And the color of the apple is a product of its unique mixture of phytochemical compounds, the chemical compounds that it produces. And if you've ever tried any of these, you know that some of them are more sweet, some are more sour. And again, that's characteristic of the variety or the genetics of that particular variety. And that's also a product of the unique chemical composition. So you can see how in an apple in particular, the genetics have an effect on the phytochemical composition. That's very obvious even just by looking at them. So the same is true of hops, and in fact of all plants, is that genetics impact the chemical composition. So how that plays out in hops, here we have two different varieties representing two different genotypes of hop, and these are the cones. So you can see they're morphologically different, and if you sort of broke them open and smelled them, you would also see that they smell distinct, perhaps if you're trained in hop smelling. And these two graphs here sort of show the aromatic characteristics of these two different varieties. So, for example, Eureka has a resinous aroma to it, whereas Cascade does not have a resinous aroma. And so again, these aromas are a product of their unique chemical composition of these hops. So here we see in hops how genetics affects the chemical composition and how that impacts some quality characteristics of this crop. And as I mentioned, this is true of all plants. The genetic composition will affect, or the genetic makeup will affect the chemical composition of that plant. So genetics, I also mentioned that environment has an effect on chemical composition of plants. So for that, we turn to wine and wine grapes. You know, as some people know, different vintages of wines, even of the same variety, like this is the Riesling variety, different vintages, wines grown, or grapes grown in different years or grown on different farms, have different aroma and flavor profiles and characteristics. So this is a chart similar to the one I showed for hops. And this is a study that researchers did on Riesling grapes grown on the same farm in 2 different years. And you can see one year in the dark color, the other year in the light color. And you can see that even between different years, the aroma and flavor characteristics are different. And that's again a result of the difference in chemical composition. And you can imagine from year to year there are differences in climate, for example. So climate is a big environmental factor. Other ones include soil types or other insects in the environment, other animals and other organisms, bacteria, fungi in the environment. Those can all affect the chemical composition of plants. So to summarize, genetics and environment are 2 of the main factors that affect the chemical composition of plants. There are a few others, such as developmental stage, post-harvest processing, and the part of the plant. For example, leaves have a different composition than fruit. So my studies didn't explicitly look at those factors, those other factors. Rather, we controlled for those other factors, or we kept them uniform so that we could just look at the effects of genetics and environment on the chemistry of our plants. So with that, I'll move into my first project. So going back to this question about how do genetics and environment affect hop chemistry, in the first project, we're asking how do wild hops from the American Southwest, species Neomexicanus, chemically differ from cultivated hops, which are mostly of European descent, species Cumulus lupulus. So, you know, when some people, and myself included, think of the American Southwest, they often think of landscapes like this. They're sort of hot and dry, desert type of landscapes. But the Southwest actually also has mountains, which sort of stand out of the desert. They're higher elevation, cooler, and in fact, this is where the hops are found. And one aspect of my project that I will not talk about today is sort of the Sky Island aspect, which Dr. Kennelly briefly mentioned. But we're following up with genetic studies to look at how different populations of hops differ genetically. But nonetheless, the hops are found in the mountains of the Southwest. And the environments of the hops are found look like this. So they're cool, and there's running water often. So contrary to what you may think, Southwestern hops are not desert hops, they're actually mountain hops. So this is a map of distribution of the genus Cumulus. So as you can see, they're distributed throughout the Northern Hemisphere and mostly in the temperate climates. So in our study, we collected wild hops from the American Southwest, this region. And all these wild hops represent the species Cumulus neomexicanus. And in comparison, and just to mention, this is one of the studies where we were looking at leaf chemistry rather than cone chemistry. So we were looking at the leaves of Cumulus neomexicanus and comparing them to leaves of Cumulus, mostly Cumulus lupulus, but a group of cultivated hops or hop cultivars. Some of these cultivars are pure Cumulus lupulus from Europe, but many of the cultivars are hybrids of European and North American hops. But one thing that all of the cultivars share in common is that they have European ancestry, and all the wild hops in our experiment are from the American Southwest. So as you can imagine, these are genetically different, but the environments that they grow in are different. So to control for environment, we grew all the hops in a greenhouse. This is our greenhouse at Lehman College, in order to control for environment. So environmental conditions were uniform for all the plants, so that any chemical differences we detect are due to differences in genetics. And the technique we use to analyze the chemical composition of these hop leaves is called Lcms Metabolomics, and I'll tell you how that works. So here we have our hop plants growing in the greenhouse at Lehman, and we sampled the leaves from these plants and made solvent extracts of them. So you can, one way to think of it is like a tea, but instead of water, we're using organic solvents. So we're making solvent extracts of these hop leaves from all of our different genotypes. These extracts are actually complex mixtures of hundreds, or in some cases thousands, of different chemical compounds. So these are just a few of the compounds found in leaves. I believe all these compounds are also found in hop cones. So we have these complex mixtures of hop compounds in these extracts, and we want to know which compounds are in it and how much of each of these are in it, right? But it's just all, it's a complex mixture, so we need some way to separate it out. So that's where the LCMS comes in. So LC stands for liquid chromatography, MS stands for mass spectrometry. So the LC part is what allows us to separate these complex mixtures into their individual components. And then the mass spec part detects the mass of these compounds to a very high accuracy, as well as measuring the mass of some of its fragments, so parts of these. And we use those data, the information, to help us identify what the compounds are. So when we run an LCMS analysis, the output looks something like this. So this is a chromatogram. And what you're seeing on the x-axis is retention time. So basically, these compounds are separated out over time. Each of these peaks corresponds to one, or in some cases, a few individual compounds. And then the height of this compound or the height of the peak gives some indication of how much is in there. So one way in which we use these type of data is to quantify specific compounds that we're interested in. Or in this case, a group of 3 compounds known as alpha acids. These are the primary bittering agent in beer. The alpha acids, they belong to a class of compounds called bitter acids. And so it's 3 individual compounds. And so what we found in our comparison was that the alpha acids in the leaves of our wild hops, seen here, were much higher than the alpha acids in the cultivated hops. So this is one use of Lcms metabolomics, is to look at specific compounds. Another thing we can do is look at overall chemical profiles and ask questions about overall how are different samples similar or different from each other. And so for that, we use a technique called principal component analysis, where for each sample, we take the measurements of all these hundreds or thousands of compounds. And then we use principal component analysis to simplify the data into its most important dimensions. So each one of these points on this graph represent an individual compound, and the color tells you which group it belongs to. So the blue ones are the cultivars, the red ones are the wild hops. And so the overall picture from this is that the cultivars seen in blue and the wild seen in red form distinct groups. So the overall chemical composition of the cultivar leaves is distinct from the overall chemical composition of our wild hop leaves. So that's what this is showing. And that takes into account all of these compounds. Another finding from our study was that there are certain classes of compounds that were more abundant in our wild hop leaves relative to the cultivars. So here are just the representatives of these compound classes. So we have the prenylflavonoids, which include potent anti-cancer and estrogenic compounds. We have the alpha acids, which, as I mentioned before, are the primary bittering agents, and the flavonol glycosides, which have antioxidant properties. And in addition to their uses to humans, these compounds are also protective of the plant. So these compounds are thought to protect against herbivory, so insects eating the leaves. And these compounds may have activity against fungal infections to the leaves. So these compounds are important to the plants as well. So these 3 compound classes were higher in our wild hop leaves. So now the flavonol glycosides are produced in the green leaf tissue. So most of what you see here is green leaf tissue. But the prenylflavonoids and the alpha acids are produced in glandular trichomes, which are specialized structures on the plant leaves. So these 2 different groups of compounds are produced in different parts of the leaf. So what are these glandular trichomes? So if we look at the underside of a hop leaf, we see all these little yellow dots. And these are the glandular trichomes. So these are specialized structures that produce certain plant metabolites, generally thought to be protective for the plant. So we also looked at the density of these glandular trichomes on the leaves of the plants in our study. And again, we're comparing the cultivar group against the wild group. And what we found is that these glands are found in much higher density in the wild hop group relative to the cultivars. And here on the right, you can see sort of the gradation of density of these glands, starting at like very sparse, getting more dense, and then very dense up here. So these glands are thought to be protective. And so there's some evidence of that. So this is from Narain and Small, did a study on hop leaves. And as you can see here, the outer parts of the leaves have been chewed away by insects. But this inner part is relatively intact. And if we looked on the back side of this leaf, what we would see is that those glands, the glandular trichomes, are found in much higher density in this middle part. So this is some evidence that these glands are protect against herbivory. Same thing in the when we look at parts of the hop flower. So these are modified leaves known as bracteoles, and and at the base of this, on the front side, on this side, we see a fruit. So a hop fruit, which is sort of a nutrient-dense fruit that holds the seed, the hop seed. And as you can see, the density of these glandular trichomes are much higher on the part of the bracteole that contains the fruit. So again, it appears to be that the trichomes, the glands, are protecting the fruit against herbivory. You know, because like I mentioned, the alpha acids, the bittering compounds, are produced in these glandular trichomes. So these trichomes are bitter. So you can imagine that being a deterrent to an animal that wants to eat the seed. So to summarize the findings of this project, the leaves of the wild hops are phytochemically distinct from the leaves of the cultivars. We saw that in the principal component analysis. We also found that bitter acids, prenylflavonoids, and flavanol glycosides are higher in the wild hop leaves, the wild Neomexicanus leaves. And this may confer improved resistance to herbivory and to disease like fungal infection. But this needs to be followed up with further study to more directly test this hypothesis. We also found that the density of those glandular trichomes was also higher in our wild hop leaves. And because the bitter acids and the prenylflavonoids are produced in these glands, this could explain why the bitter acids and prenylflavonoids are higher in the wild hop leaves, and it likely does. You know, basically they have more glands, so they have more of these compounds. As Dr. Connelly mentioned, this was just accepted this week in the Journal of Agricultural and Food Chemistry. And our findings were also, our field work was covered in some popular press and in various magazine articles. Most recently, a book out of Cornell University Press interviewed me for a little segment of this book about how climate change is changing the crops that we love. So that was nice. So if you're curious to learn more about the field work, which I didn't really talk about much in this talk today, you can go to my YouTube channel. So the name of my channel is just Taylan Morcol. It's just my name. And I have several videos uploaded from my field work in the American Southwest. So there's a mystical quest for an elusive wild hop. There's a flash flood that surprised us. So you can check these videos out on my channel if you like. There's a wild hop playlist. If you click on playlist, that has all the wild hop videos. And also the recording of this talk today will also be uploaded to this channel. So Taylan Morcol is the name of the channel. Feel free to check it out. So with that, I will move into my second project, the drought experiment. So again, getting back to the central question of genetics and environment and their effect on hop chemistry. In this study, we're looking at both genetics and environment. And so the environmental aspect was drought. So we did different drought treatments. And we're testing differences in genetics because we use multiple genotypes. So this experiment was also conducted in the Lehman College greenhouse. And Conrad Wysocki, a former master's student who's now doing a PhD at Rutgers, was a huge part of this study. So this was actually part of his master's work as well. So in this experiment, we had 3 different treatment groups. So we exposed the plants to 3 different treatment groups, basically just different amounts of water. So a lot of water, less water, less water. And we replicated these treatment groups across 4 different genotypes of hops. So there were 3 commercial cultivars and 1 of our wild Neomexicanus hops. And we used a very similar technique of Lcms Metabolomics to profile the chemical composition of the leaves. So again, we were looking at leaves here because we wanted to understand how the drought stress was affecting the physiology and metabolism of the leaves. So we're looking at questions of basic biology here. Again, we use principal component analysis. In this particular figure, we have a different plot for each of the 4 genotypes in this experiment. So each point represents one particular plant that we sampled. And the colors represent the treatment groups. So blue is the control group that received the most water, all the way down to red, the severe drought group, which received the least amount of water. And what we noticed is that, so in this, remember, we're looking at sort of the overall chemical composition of the leaf. And what we found is that there's a tendency that, for example, in this case, the plants in the severe treatment group that received the least amount of water were chemically distinct from the leaves that received the most amount of water. This trend was most pronounced in the Cascade cultivar, but there is also a similar trend, although not as distinct, with the Neomexicanus, with the Sultana, where red is up here, blue is down here. Not so distinct with Aurora. But overall, it did seem like our drought treatments produced a pronounced and noticeable effect on the chemical composition of these hop leaves. So this is looking at each genotype individually. But when we zoom out and look at all of them together on the same plot, we see that each of these genotypes is pretty distinct on the PCA. And so, for example, this is the Sultana genotype, and these are all of the different treatment groups together. The control, severe, and moderate are all here. So this, generally what this is telling us is that even though the drought treatments did have some effect on the overall composition of the leaves, genetics appeared to have a much larger effect. So even though we impose different drought treatments, each genotype, the chemical composition of each genotype remained distinct overall. So we also asked the question, what specific compounds were most affected by these drought treatments? And so here in this figure, we see some of the compounds that were consistently affected by the drought treatments. So the arrow next to each of the compound names indicates whether it increased or decreased in response to drought stress. So whether it was higher in the severe treatment group or lower in the severe treatment group. So with the exception of this compound, they were all higher in the drought treated plants. And a lot of these compounds are known signaling compounds, so they're involved in the drought stress response. So here we have lipids, and for any chemists in the group, these are the polar head groups, and then the fatty acid chains are represented by R groups. So all these lipids are likely, they're all monoacylated, and they're likely signaling molecules, and they're all breakdown products of the cell membrane. So membrane breakdown is known to occur in drought stress plants. This compound is a breakdown product of chlorophyll, and it's also involved in signaling in drought stress. These two are breakdown products of carotenoids, and abscisic acid is a very well-known plant hormone. So these, generally, most of these compounds were very consistent with what is known about drought stress. A few of them, glutaric acid, dihydroamericidin, we're not quite sure, just based on the literature, why these compounds were consistently increased or decreased in response to drought. So that is sort of a question that remains unanswered. So to summarize the findings of this project, the leaves of each genotype were phytochemically distinct. Now, despite the drought treatment effects, yet the drought treatment effects still did have some noticeable effect on the overall metabolite composition, as well as on specific compounds, which served as evidence of not only breakdown of different components of the plant cells, but also are involved in signaling, so in stress signaling in plants. And again, as Dr. Connelly mentioned, we published these in Journal of Agricultural and Food Chemistry last year. This is the team that was involved in that publication. So with that, I'll turn to the third project, the Terroir study on a class of compounds called Aromaglycosides. So again, we return to our question of genetics and environment and how they affect hop chemistry. And in this particular project, we're growing hops in different locations that have different environments. We also grew various multiple genotypes, like we did in the drought study. So we have multiple genotypes in different locations. So we're testing both of these factors and their effects on hop chemistry. In this particular study, we're using hop cones instead of leaves. So what is terroir? So the way this experiment was set up was to grow hops, 23 genotypes, the same 23, in 2 different locations. So Yakima and Kootenai. So this is a map of the Pacific Northwest region of the United States, where most of the hops are grown in the US. Yakima Valley is a high desert environment, and this is where the majority of hops in the United States are grown. So we have the Yakima Valley here, which is high desert, extreme temperatures. And then we also have the Kootenai River Valley here, which is in northern Idaho. It has more moderate temperature, higher humidity. And the soils between these 2 locations are also different. So all these factors, climate, soil, etc., define a particular environment's terroir. So in this case, we're using the term terroir to mean the set of environmental conditions that are unique to a specific location. So we have 23 genotypes growing in 2 distinct locations that have distinct terroirs. Now the Kootenai hops were grown on a farm called Elk Mountain Farms. So just for fun, sometimes we like to refer to this project as the Elk versus Yak project. So as I mentioned in this project, we were quantifying a class of compounds called Aromaglycosides. So unlike the previous projects that I talked about, in this project, we were only looking at 4 specific compounds. And so what does aromaglycoside mean? So the aroma part corresponds to, in this particular case, this part of the molecule here. And glycoside is this part, so it's a sugar molecule, where the word glycoside comes from. So all these molecules are a volatile aromatic molecule bound to a sugar molecule. So we call them, in this case, aromaglycosides. And so when these compounds, when the aroma part, the volatile part, is separated from the sugar, each of them has a different distinct aroma. So some of them are grassy, raspberry, floral, honey, spicy. So each of these compounds has sort of a unique aroma. In plants, these volatile compounds are often stored in their glycoside form for various reasons. One main reason is that the sugar makes this molecule more soluble in water, so it's easier to store and transport. So plants will often store these sort of volatile parts and aromatic parts, which are often the bioactive part, whether it be in signaling or defense. They store this sort of volatile bioactive part bound to a sugar, and then they can kind of release them upon demand by cutting this bond. So we measure these 4 compounds in hop cones from grown in those 2 locations. And so each of these graphs represents our measurements of each one of those 4 compounds. So abbreviated here is raspberry ketone, 2-phenylethyl, hexyl, and linalool. So we measured the relative quantities of these compounds in all 23 varieties and in 2 locations. So locations represented by different colors here. This is a lot of data. So to make sense of it all and to look at the question of what is the effect of the genetics and the environment on the levels of these compounds, we performed analysis of variance, also known as ANOVA, to look at basically the significance and the size of the effect of location, which is our environmental factor, and cultivar or genotype, which is our genetic factor. And what we noticed, this stood out to us. So this compound, the hexoglucoside, even if you look here, you notice that for each of these pairs of bars, the red bar is higher than the blue bar, which indicates that the sample from Yakima had a higher level of this compound than its corresponding sample from Kootenai. We thought that was pretty interesting. So what the analysis of variance shows is that, in general, this column that's highlighted here is a percentage, and it shows the size of the effect of this particular factor on the levels of a particular compound. And in this case, we found that location accounted for almost 50% of the variability in the level of the hexoglucoside. So almost 50%. And if you look at all the other compounds, it's much smaller, less than 1%, 2%, less than 1%. But so location had a huge effect on the levels of this compound, but all of the other compounds were primarily affected by genetics. So in these two cases, almost 95% of the variability in these compounds were affected by variation in cultivar. So we thought that was interesting. We kind of asked the question, like, well, why is that? What does this mean? So looking at this compound, the hexoglucoside, the aglycone part, or the volatile part by itself, is 1-hexanol, and it belongs to a class of compounds called green leaf volatiles. So these are the compounds that are released when a plant is cut or damaged, and they contribute to that fresh cut grass smell. And they're released within seconds of the plant being damaged. So if you're familiar with the smell of fresh cut grass, you're smelling this class of compounds, these green leaf volatiles. So these compounds are released when the plant is cut. Also when herbivores are eating, so when herbivores start to damage plant tissue, these compounds are also released. So this is a close-up of a hop leaf infested with spider bites. And so what we suspect in our experiment is that the Yakima environment, the one that had much higher levels of these compounds, of this compound, had much higher levels of insect pressure. So there's a lot more herbivory going on in Yakima. And although the design of our experiment didn't allow us to hone in on one particular environmental condition, we suspect that herbivory may be a big factor contributing to the higher levels of this compound in our Yakima samples. So the main findings from this project, we found that genotype had a substantial effect on the levels of all 4 of the aromaglycosides. Actually, even on the hexoglucoside, genotype did play a role in the levels of that compound as well, and certainly of all the other compounds. But location was the dominant factor affecting the levels of hexoglucoside, that green leaf volatile glucoside compound. So location was the main factor driving the levels of the hexoglucoside compound. And this may be due to insect pressure. In reality, there are other stressors that can increase the accumulation of green leaf volatiles and their glycosides. So again, this would need further study to test this hypothesis. And these findings were also published in the journal Food Chemistry last year. And then this article was covered in a Science News article, sort of a popular science magazine, along with some other studies looking at terroir. So this was a really nice study. I think they also looked at grapes and coffee and things like that. So this was a really nice bonus. So with that, I want to just talk about some of the main conclusions overall from my work. So what I found in my studies is that genetics seems to play a major role in determining the phytochemical composition of hops, both in the leaves and in the cones. Still, environmental conditions do have some effects, and in overall, certainly in the overall chemical composition, as well as more substantial effect for individual compounds, like we saw with the hexaglucoside with that green leaf volatile. And Lcms Metabolomics is a powerful tool, not only for studying plant metabolism, so like basic biology questions, but also for assessing the crop quality. Because as I mentioned, a lot of the compounds that we were measuring contribute to the quality characteristics of hops, such as their bitterness and aroma. So with that, I want to just end with some acknowledgements, because there are so many people that contributed to the success of this project or these projects. So I want to thank my committee members, Dr. Paul Matthews at Hopsteiner. As Dr. Kootenai mentioned, he's been a huge part of not only funding this project, but sort of directing the vision, and then the Wild Hop project was Wild Hop Conservation Project is Dr. Matthews' brainchild. Dr. Damon Little at the New York Botanical Garden, who is expert in population genetics, and I don't really have much experience with genetics, so his experience and perspective on the genetics and also statistics were really valuable for my project. Dr. Renuka Sankaran, Lehman College, is a plant physiologist, and her expertise on plant physiology was really valuable in designing the drought experiment especially. And she's provided a lot of critical feedback in terms of just experimental design and really making sure that the experimental design is solid. And then Dr. Michael Balick at the New York Botanical Garden was a world famous ethnobotanist. And Dr. Balick's perspective on sort of the relationship between plants, people, and culture has really helped me understand my research in the context of culture and human history and economics. So I really appreciate Dr. Balick's perspective as well. So several institutions also contributed to this project. As I mentioned, Hopsteiner, who's directed this research, Dr. Paul Matthews' team, as well as provided a lot of funding. I'm really fortunate in that in the last almost 6 years of my grad school, I didn't have to teach a single class, and all of my support came from Hopsteiner, which really allowed me to focus fully on my research. So I'm really grateful to that. And a special thank you to Harold Schwartz. New York Botanical Garden, as I mentioned, two of my advisors are from the garden, and several other people were helpful there in learning GIS, and Valisa Vasquez and Michelle Misawan for really helping me with a lot of the administrative stuff. So many people to thank at Lehman College, where I spent most of my time during my grad school. Dr. Dwight Kincaid, I took a biostatistics class from him a couple years ago, and it was just so incredibly fun and so useful to my project. And he's really helped advise parts of my project as well. When I have questions about statistics, I'll go to Dr. Kincaid or Dr. Damon Little, as I mentioned earlier. And the administrative team, Patricia Carver and Dolores Vatanza, just helping me take care of so many of the administrative things. It's just so important to navigating through the system. So I really appreciate everyone on here, and yeah, thank you. At the Graduate Center, Miss Joan Reed, every grad student at every PhD student at CUNY comes to appreciate Miss Reed for all the administrative help that she provides. Will McGowan, Debbie Sacco, or a few of the other ones that really helped a lot. And People at Waters Corporation, the company that makes our LCMS instruments. In terms of operating these instruments and learning about them and fixing them, I just learned so much from their technicians, both on site and over the phone. I spent so many hours learning from them, and a lot of what I learned in LCMS is thanks to these people. At the USDA Agricultural Research Service, especially Pat Reeves and Dr. Chris Richards, who were really instrumental in designing the wild hop part of the project, especially. So yeah, a big thank you to them. In the Kootenai lab, I just want to mention a few people. Ella Vardaman, who's one of my co-hosts today, I really thank you, Ella. Dr. Adam Negrin, who was a PhD student when I first entered the lab, was really a big mentor early on in the lab, and also a great friend. He was just, Adam was so generous with his time and hospitality, letting me stay at his place. I really appreciate Adam. And also Dr. Adam Cavalier, who was the previous hop student in my in the Kootenai lab that a lot of my research is built upon. So thank you, Dr. Cavalier, as well. At the USDA Forest Service, so I did most of my field work in national forests. And there were a few people here that were just so generous, especially Larry Cordova and his team, Dakota Crane and her colleagues, not only providing like field support during collections, but also like helping me with accommodations, so I didn't have to sleep in a tent the whole time. I really appreciate the Forest Service as well. A lot of other people to thank here. Some of my former colleagues at the Pennsylvania Biotechnology Center who are here today. I think Dr. Kinney is here, Dr. Getz, Dr. Block. Dr. Getz and Dr. Block especially were really encouraging me to do a PhD when I was there. And I learned a lot of my sort of phytochemistry lab practices early on from Dr. Getz, so I really appreciate his mentorship as well. And last but not least, Dr. Kennelly. Dr. Kennelly has been my main mentor throughout my PhD studies. He has taught me so much, not only about phytochemistry, but also how to be an independent researcher, how to navigate the system, how to write emails. And I've come to deeply appreciate his mentorship. So thank you so much, Dr. Kennelly.